was just completely crazy idea, which didn't really succeeded. But anyway, and so during my PhD, I was working for plant physiology as assistant feature editor, and was also grad school representative at that time. So I was not sure how this is going to work today. So I tried to just to raise some bullet points, and I thought you had enough input today by all the talks. So I was not so intrigued by just giving another presentation on our paper or on the work I did in my PhD. So I think it would be also for you more valuable that we have some kind of a discussion. So I'm not sure, maybe so I can just comment on what I'm now doing. So after I graduated in December last year, I joined now Bayer, and now I'm working in Bayer CropScience. So I think that iGEM is a very powerful and also very nice competition. So it's a lot of fun to connect with all the people across the globe. And I just still, since 2016, I just follow it as a side pass. And I think it's amazing the projects you see there. And it's amazing how creative people are and also how creative you are. Whenever you have questions or you think there's something you want to ask, I will drop my mail in the chat. So just write me an email, maybe give me 2 days to respond. And also because I also switch topics a little bit. So sometimes I might need to spend 1 day reading on it so I can make a comment on it. Okay, so just feel free to reach out to me. So in my PhD, I said I did a lot of metabolic engineering, so I could help you a little bit with cloning in plants, choosing promoters, terminators, kits, cloning kits you want to use, also on transformation. So I did a lot on transformation. What Jen explained on using tobacco, using Arabidopsis, and then in addition, I also did quite a lot of analytics, so really metabolomics, so all the stuff with mass spectrometry. So this is basically what I can provide, and in Toby's lab we also did some enzyme tests. So yeah, I think it's fine. Maybe I'm just curious to hear. So what are the expectations you have to me? So what would you like to know? What should we discuss? I think 40 minutes is a very limited amount of time also to pitch the project. So I'm just curious to know. And also to know on the project you're going to work on. If you're free to share, I mean.
Yeah, so I would have a short question, I guess. So since you do have a lot of experience with plants and also different kind of plants, I'm always just wondering, in like your experience, what the most limiting step is in all these plant transformations, because there's still like a lot of, I mean, you can do some transformations in some plants, but there's still many more that are not really transformable. Like where do you see the big issues here?
So, I mean, the transformation efficiency in plants is like super low. And I mean, this is an issue that a lot of people are working on. And I think that's something with a limited amount in an iGEM competition, it's nothing that can really be solved. So what I think, or what I would like to give you also as an advice is, because in the beginning you will generate a lot of genetic parts and you also need to test it. So I think try to think about a system that allows you to rapidly test all the things you are going to clone. And I think then the transient transformation in tobacco is a super nice system, and there are a lot of protocols out for also protoplasting. So I think to work in the beginning with protoplast to test your systems, use fluorescent reporters, I think that's something that has a high value. And once you have established a really good construct or something where you think, oh wow, this is good, then you can still try to transform an Arabidopsis plant. And I think that's the most important advice I would get. And the other one is always think about, when you design your system, to have an easy readout, because you can test a lot of things, but if your readout takes you 1 week to prepare lysates and stuff, then it's not fast, and then you cannot test a lot of stuff. So I think to spend 1 or 2 days more on planning this, or maybe to split it into 2 different systems, a test system with an easy readout, and then like a higher level tests with a proper readout, where you also put more effort in elucidating what you want to test. I think that's really a key to succeed in a plant iGEM project.
Yeah, thanks. It's definitely true. So for these kinds of let's say transient essays that you can do to kind of quickly characterize something. So Jennifer Brophy earlier mentioned that there can be quite some issues if you have, because you have like different effects in different tissues, and the expression might be different. So did you have in your work also problems like this, where you tested something in like your fast prototyping system, and when you actually did the real transformation, it didn't work at all?
I think there, how am I going to phrase the answer? It's a very good question. I mean, the point is, when you are working with tobacco, the most elegant way, and I think this is what also Jen did, is to have a reporter on your thing, because then basically you can normalize everything to your reporter, and you have a very stable readout that basically corrects for all the noise you have in a transient system. This is a thing I would advise for a transient system. Nevertheless, even if you have a system or you have a construct which perfectly works well in a transient system, it can fail when you try to put it in Arabidopsis. And I also had the same, that I had a super nicely designed pathway that was perfectly expressed in tobacco, but I never succeeded to put it into the genome. And just the point is, when you are doing the transient assay, your tDNA, so I guess you know what a tDNA is. Okay. So your tDNA is not fully integrated into the genome. And when you are trying to put it in Arabidopsis, of course, it has to integrate into the genome. And there are a lot of things that are not really known, so which is also tDNA instability. And these are things that are now raised, because like making this huge construct is so important now to really engineer a complex trait. And the advice I would give, if you have a construct which you want to put in Arabidopsis, try to get 2 or 3 constructs and do everything in parallel. In general, the more you test, the more you clone, the more you try to do, it always increases your chances to really succeed.
Yeah.
I mean, I know it's more effort, but if you come to the point where you want to put it into the plant and you realize that your construct is, or one of your construct, it's not working, then yeah, I think it's not very good for the project. And the last point I would like to say is, maybe, I know that iGEM is super exciting. Yeah, and all of you have, I'm pretty sure, have amazing ideas and things that are interesting. But if you want to have, or really get a nice reward in iGEM, try to not think what is interesting, try to think what is good for the project. How can I push the project, and not what is interesting for science? I mean, there's a lot of interesting stuff in science. Everything is interesting, and it's super fascinating what you can do now. But some of the things don't really help you for your project. And then, so this is in the end how you get graded by the iGEM judges. So what is good for the project should always be the first question before what is interesting.
Yeah, that's a very good point. I mean, this always happens. Like someone comes up with a super crazy idea. Oh, this would be so nice to test, but in the end, you only have so much time.
Completely agreed. Yeah.
Yeah. Thanks for your answers.
I had actually some questions, if it's okay. So, when you do plant transformation and you obviously check, there's in your construct there's a promoter, there's an UTR, there everything is in frame, so the obvious thing. What are like mistakes you don't think often of, or you that are easy to overlook, that might be part of it?
So I think that when you design the construct, there are a lot of tools out. Yeah, a lot of like cloning boxes that have promoters, that have UTRs. And I think the experience that I made is it's always better to use plant-specific promoters than using an agrobacterium one, than using a 35S, or using a NOS promoter and all these bacterial promoters, because you always have the issue with silencing. And silencing in plants is really a thing. So plants don't want to express anything that is not in their genome naturally. And they do everything to silence it. But once you have so try to use Arabidopsis specific promoters. There are a lot of strong promoters described, like ubiquitin 10, and you can just drop me an email and I will just send you back some promoter names. If you want to only work in photosynthetic tissue, you can use Rubisco promoters, because then you have the advantage that the plant cannot silence this promoter, because otherwise it will completely ruin its own life cycle. And then there is a very nice paper, which is from Nicola Patron. So if you haven't reached out to her, I really recommend to do it, because she's one of the amazing scientists I ever talked to. And she had a very nice paper on designing minimal synthetic promoters for plants. And the paper came out when I was already writing up and had all the constructs done, but I think that's also super interesting. I'm not sure if you are aware of the paper, but just put it in the mail and I will send it back to you.
Cool. On that note, actually, I have two more questions. First, inducible promoters for in vivo plants. I mean, I found some. I think it was a sugar promoter or something like that. And the second one, the selection marker. I know that recently people use like these Basta resistance or PFAST, which is like a seedling promoter for GFP or something like that. What are your experience on that front?
So I used, I would say, all of it. The best experience I made, so kanamycin works very good as a selection marker. I think in most of the standard cloning kits there are also kanamycin resistance cassette pre-clone, so it's super easy to handle these. Hygromycin also works, but hygromycin is super toxic for the plant. So if you have, for example, a strong phenotype or something, I don't know. I have some experience that I made with expressing this GFP seed-specific GFPs. Mostly, I did it when I was doing CRISPRing in Arabidopsis, and it's absolutely amazing. So if, as a recommendation, I would either choose kanamycin and also seed-specific expressed GFP.
Okay, thank you.
The last question, I think you had.
I know this promoter stuff.
Okay, good. Yeah.
Okay.
I maybe have another question. So, what's like? Do you have experience with tobacco?
I did only transient expression, so no one in our lab really worked in real tobacco. But as far So we always had the idea, but then it didn't run out because we had everything set up. So are you planning to do something in tobacco or?
Yes, and we wanted to detect a chemical which has to be first absorbed or uptaken by the roots, and we asking ourselves how long will it take to from the roots to the leaf? So in which period of time? Do you have any experience on that?
So I mean, it depends. I would say it depends on the chemical.
If it's polar or unpolar.
Of course. So uptake is always a problem, but that's something that can be easily solved. So what you can do is it's called a stable isotope labeled metabolite. Yeah. So basically, you have a mass of +1, and that you can easily detect and distinguish from all the native stuff. I think it's fast. So I was doing labeling experiments where I was feeding plants with stable isotope labeled glycine and glutamate and serine, and then I distinguished how much is left in the roots and how much do I actually detect in the shoot, and usually the uptakes works extremely well. Great. I did it in Arabidopsis, but I think in tobacco it is not different, and in the end it all depends on the gradient you have. So when you apply a lot at the root, you have a strong gradient and the plant will automatically transport everything to the shoot. So what are you aiming to apply? Is it a chemical or a metabolite that is already in plants, or?
No, it's hopefully not already in plants. It's like a toxic chemical.
Okay.
And then we want to detect them. So like if the ground is contaminated with it, that's somehow our system.
So the idea is that the tobacco plant, which is grown on the field, basically recognizes the contamination in soil.
Yes.
That's amazing.
Basically.
That's amazing. I think it's, and then you have like a reporter in the leaves, which is glowing. Or what?
We thought about rubies, so the plant gets red. Or we could also use different, but that's the main one we want to use.
That's a pretty it's a cool super cool idea, and I mean this recognizing things in soil. So I now switch to soil in Bayer, and it's cool stuff. I like it. So I can contribute with both expertise, makes it even better. Anyway, so I think for the toxic chemical, I mean, just briefly test how toxic it is. So how much you can apply in the soil. But usually I think that's something you could work, and I'm not sure if you have access to analytics, but in principle you can detect everything. You can also find everything in if you have proper mass spectrometry access, you can find everything even at low quantities. But so I think it's fine. And I mean, if you cannot get a stable isotope, you can also look for radioactive labeled one. If you have a radioactive one, that you can also detect them.
Yes, it's not about that. We are not able to detect the chemicals, but it's about to detect it in huge areas, so we don't have to probe it everywhere, because the plants will grow everywhere, and then we can fast detect where the chemical is, which regions are contaminated. That's the plan.
And I mean, for tobacco, tobacco also works with agrobacteria, the transformation, and I think it's more like putting a leaf on agar medium and add agrobacterium and wait, and then you have this callus formation. They are like super old protocols, and in general look at the old protocols because they usually work better than the new ones.
Thank you. We wanted to do it with Arabidopsis, but then we saw that we have limited time in iGEM, and then we just tried now into tobacco.
I think it's a good decision. I mean, I'm not sure. I mean, for Bentamiana, I think you can also do it with a protoplast first. Yeah, just to see if your reporter constructs are working.
We will.
Good. I mean, then you're good. You're good to go.
I have a follow-up question on this. Why exactly do we need the stable isotope thing if we do not have this chemical already in plants? Can't we just detect?
Yeah, if you don't
phenomenon?
have it, yeah, if you don't, because if it's not natural to plants, then you of course don't need a label.
Okay, thanks.
That
Yeah,
makes
I
it-
think that this makes it a bit easier.
Yeah. It's just if you have something that probably is there in plants, then you can still distinguish it by that.
Okay, makes sense. Yeah.
For example, we would have to label arsane and stuff because it's natural in plants.
Mm-hmm. No,
But
that's
we won't do that.
no, no. I think that's okay. Good. Further questions?
Yeah, about protoplasts, if you know anything about that. How to transform them best, especially when we want to stably transform them.
I think there are two ways how you could do it. The first one is you infiltrate the leaf, incubate it for 2 days. So you have it on a whole leaf, incubate it, and then isolate after 2 days. You isolate a bunch of protoplast and do some analysis with the protoplast. Protoplast and protoplasts in general are not very stable. So I think they're just stable for 1 or 2 days. So this is something where you have to test it quite rapidly. And the other thing you could do is you take an untransformed leaf, and then there is a method, it's called PEG-mediated transformation of protoplast. And thereby you basically have like some kind of chemical, and this carries and the purified tDNA plasmid into the protoplast, and then you have really do a batch of protoplast. But that also works, and there are also publications out that describe this how this works. And I think this is the two ways how you could get transformed protoplasts, but they're really not stable. It's really hard, so you need some practice to handle these because they break quite fast.
Okay, thanks.
Yeah, I think we still have time for another question.
So I have a question. I don't know if you know too much about tomato and agricultural, but we are working with a project on pesticides and tomato. So we are thinking about preserving the life of bees, and we are aiming to degrade pesticides specific to pollen. So my question is, do you know if how much the plant is able to absorb pesticides from applying the outside? Because normally they apply on the seed and they can pulverize it again later, but we haven't found data about the pulverization, how much it absorbs, right? And also, if you know some kind of method that we can measure the pesticide quantity in Poland.
In Poland?
Yeah.
So to your first question, for the absorption of the pesticide, the extent to which a pesticide absorbs to the plant depends on the application rate.
Mm-hmm. Yes.
And also on the chemical properties of the pesticide. And so I think to have a general value, I think this does not exist. Or basically, like if you apply a pesticide to a plant, then 90% of the pesticide are adsorbed to the plant. I think that doesn't exist, because it depends on the plant. It depends on also the density of the planting, and also on the chemical. If you you can have a look. It's called the pesticide database or something, and there you can look for specific chemistry that is applied to tomato or something. Yeah, and there you always I'm pretty sure you also get values on absorption or things like that.
Okay.
And for the first question, the second question, which I think was if you have a method to detect pesticide that remains in the pollen, is that right?
Yeah.
I mean, in theory, what you could do is you need to harvest all the pollen, and then you need to do an extraction of the pollen to isolate the pesticide, and then do an LC-MS measurement, and then you can quantify how much pesticide you have if you know which pesticide was applied.
Okay.
Or you can also do either you do it via a mass spectrometer, or if you have a high enough application rate, you can also do it via an HPLC measurement.
But since it's a small quantity, do you think we will need like a lot of plants to get a good result? Or
so I mean, what is small quantity? Is it
because we're taking specific to the pollen, right? So the pollen is very little.
I think if it is higher than one, I mean, in theory, try to give it a calculation, and we can also set up like a call to do it together. If you assume a certain application rate for an area of field, and then you could assume a certain amount of plants that are on it, and then basically narrow it down from the application rate, and then you can at least get a guess. If it is higher, then so if you have on one plant, you have maybe 1 nanogram of pesticide in the pollen, I'm pretty sure you can still detect it.
Oh, okay. Yeah.
So if you do it with a mass spectrometer, it's so sensitive that you can still detect it.
Okay. Thank you very much.
Yeah, welcome.
Okay. I think this session was really productive. A few really interesting questions, and thank you very much, Marc, for joining and helping us out with the plants and stuff.
You're welcome.
So now I think we could go back to the main session, main Zoom meeting.
Yeah.
For the team elevator pitches, which I am very excited to see. So
good. Bye. Feel free to contact me if you have questions. Yeah. So don't be afraid.
Thank you very much for joining.
It just may take 1 or 2 more days, because my current employee, because I will do this in my free time. So, but I'm happy to help. Yeah.
Thank you very
good.
much.
All right. So how are we coming back now? Is it like someone is transitioning us, or?
And on the bottom right corner, you see leave room in blue.
Yeah.
You click there and then click on leave breakout room.
Thanks.
Recording in progress.